Epigallocatechin gallate is a green tea flavonoid and it has known anti-cancer, antioxidant and anti-inflammatory activity. In lab studies that used human glioblastoma cell lines, the exposure to the EGCG contributed to cell death. The EGCG targets several cellular events that are mediated by matrix metalloproteinases, including some of the pathways that control cellular migration. The EGCG can also inhibit a protein that makes glioblastoma cells more resistant to chemotherapy, and it blocks their death. In human glioblastoma stem-like cells, the EGCG synergized with the effects of temozolomide. A study found that this synergy might be due to the reversal of the resistance mechanism, namely MGMT, within the glioblastoma cells. EGCG and other catechins from the green tea might fight cancer partly through their ability to inhibit the activity of an important cellular signaling pathway. In two different human glioblastoma cell types, the EGCG activated cell death pathways. Interestingly, the EGCG did not have this effect on healthy human brain cells. One study showed the green tea led to senescence of the glioblastoma cells without harming the normal cells. Another cell study suggested that the dose derived from drinking the green tea may prevent brain tumor development, but glioblastoma cell death is only achieved from a much higher concentration of the green tea. Research in mice with glioblastoma is also encouraging. The EGCG significantly improved the therapeutic effect of temozolomide, and the combination extended survival of the mice when compared to temozolomide alone. In a separate study in mice who were implanted with human gliomas, the EGCG slowed the tumor growth by interfering with the aberrant metabolic pathway of the gliomas. Interestingly, the application of the EGCG to colon cancer cells harboring a mutation in IDH1 led to a decreased proliferation. Whether this will hold true for IDH1 mutated gliomas has yet to be studied. Chrysin is a naturally occurring flavonoid and is found in honey, propolis and many other plants. It may fight inflammation and other processes that are involved in cancer development. The chrysin promoted cell death in studies on several glioblastoma cell lines. Another study found the chrysin reduced the mitochondrial function of the glioblastoma cells, and it decreased the production of a protein that's involved in tumor invasion. In a mouse xenograft model, chrysin inhibited the proliferation, the migration, and the invasion of glioblastoma cells, and it suppressed tumor growth. In an in vitro study, an extract of propolis inhibited the growth of human glioblastoma cells in a dose and time-dependent manner, and it enhanced the effects of temozolomide. Another plant-derived compound called apigenin inhibited the cellular pathways that are involved in glioblastoma cell proliferation and survival. The apigenin treatment caused the cells to stop at a certain point in their cell division process. The apigenin also powerfully suppressed the invasiveness of glioblastoma stem-like cells. This is a significant finding because the stem-like cells can self-renew and they're resistant to radiotherapy and chemotherapy. In human glioma cells, the apigenin reduced the production of TGF-beta1, a signaling molecule that's involved in migration, invasion, and the formation of blood vessels. Importantly, the apigenin might not have the same effects on normal cells. One study found apigenin activated cell death pathways in two different human glioblastoma cell lines, but not in normal human astrocytes. Central to any cancer growth is their ability to evade the immune destruction. And the apigenin was able to reduce the glioma cell migration and to restore the immune attraction towards the glioma cells, a necessary component of their eventual destruction. The apigenin has numerous anti-cancer actions across a diverse array of tumours. Phytoestrogens are plant compounds and they're similar in structure to the hormone estrogen. Soybeans, flaxseed and nuts are all good sources. Despite their similarities, the phytoestrogens appear to play a different role in glioblastomas than the estrogens naturally produced in the body. This might be because they act differently inside the cells. Endogenous estrogens have unpredictable effects on glioblastomas, leading either to their growth or their inhibition. This depends on numerous factors. However, in contrast, the plant-derived phytoestrogens have been shown to exert beneficial effects in glioblastoma. In a mouse model of human glioblastoma, a phytoestrogen called genistin inhibited the tumor growth after 10 days of treatment. Cellular and molecular analysis suggests the genistin slowed the tumor growth by decreasing the formation of new blood vessels in the tumor. Another study found the genistin may decrease the proliferation of the glioblastoma cells by stopping their division and lowering the activity of telomerase, an enzyme that cancer cells need to protect the ends of their chromosomes and to survive. In a preclinical experiment, the genistin inhibited a signaling pathway used by the glioblastoma cells, even at very low concentrations. The glioblastoma cells that were exposed to the genistin had more sensitivity to the killing capacity of the therapeutic radiation doses, lowering the dose needed to kill the cells. In another experiment, the genistin lessened the invasive potential of the glioblastoma cells. Didzin is another phytoestrogen, and one study found that didzin can help activate cellular pathways that are involved in cell death in the glioblastoma cells. Healthy brain cells were not affected by this treatment. A lesser-known phytoestrogen, called biotin A, found in red clover and other plants, also enhanced the anti-cancer effects of the thiazolamide. Hanekiol is a natural bioactive polyphenol, and it's extracted from the bark of the tree Magnolia officinalis. It's demonstrated anti-inflammatory, antimicrobial, and anti-cancer effects in lab studies. Researchers have reported the hanekiol can inhibit the division of the glioblastoma cells and cancer stem cell lines, and it can kill glioblastoma cells by several mechanisms. The ability of the hanekiol to cause glioblastoma cell death might result, at least in part, from its ability to stimulate a protein that causes cell death and inhibits a protein that prevents cell death. Another study found the hanekiol inhibited the interaction between human glioblastoma cells and cells that line the blood vessels, suggesting it might inhibit the spread of the tumor cells via the bloodstream. In glioblastoma cell culture experiments, the hanekiol and a similar compound called magnolol were more effective at killing the cancer when used together. Hanekiol is of particular interest for the treatment of glioblastomas because the studies in mice suggest the compound can cross the blood-brain barrier. In a mouse model of human glioblastoma, the hanekiol caused cell death, and it significantly prolonged the survival of the mice. A number of genes involved in the regulating of the cell cycle were activated in the treated mice. In a similar study, the combination of hanekiol and magnolol inhibited the tumor progression, and it killed the cancer cells more effectively than the chemotherapy drug temozolomide. In an experiment using hanekiol and a disulfiram copper complex in a liposomal delivery system, the tumor regression via the immune attack was observed in a mouse model. When combined with temozolomide, several studies have shown the hanekiol may complement the drug's ability to kill the glioblastoma cells. Several types of polyunsaturated fatty acids have been studied for the treatment of glioblastoma. Treating glioblastoma cells with DHA, an omega-3 polyunsaturated fatty acid, led to several cellular and molecular changes that indicate cell death. The authors followed up with an additional experiment in mice with glioblastomas. The mice were altered to express an enzyme that converts the omega-6 polyunsaturated fatty acids to omega-3, and the increase in the omega-3 was associated with a decrease in tumor volume. When various types of glioma cells were exposed to different polyunsaturated fatty acids, including arachidonic acid, gamma-linoleic acid, and DHA, the expression of certain genes involved in the cell death increased. Open-label clinical studies have suggested the GLA might be effective against malignant gliomas. In patients with gliomas, delivering GLA directly into the tumour was found to be safe, and in some cases led to tumour regression. Several participants survived without new symptoms for up to 2 years. Silibanin is a biologically active compound, and it's extracted from the seeds of milk thistle. It's capable of affecting many of the classical characteristics or the hallmarks of cancer that allow the cells to grow and to spread unchecked. Studies show that it caused glioblastoma cells to enter into self-destructive mode through at least 2 separate mechanisms. In another study, it inhibited the invasive features of highly invasive glioblastoma cells. Another strategy tested it in combination with luteolin, another plant-derived compound. The combination inhibited the growth of glioblastoma cells more effectively than thiazolamide. It slowed cell migration, and it caused the glioblastoma cells and the glioblastoma stem cells to die. Using several different glioma cell lines, the milk thistle increased the toxicity of the thiazolamide on the cells, improving the drug's cell-killing ability. The Silibanin also worked well in combination with arsenic trioxide, a drug that's approved for a treatment for leukemia. In glioblastoma cells, the combination of the Silibanin with arsenic trioxide slowed the tumour cell metabolism, and it increased the cell death. A study found the milk thistle increased the accumulation of arsenic inside the glioblastoma cell treated with the arsenic trioxide. When the milk thistle was combined with Chrysin, arsenic trioxide levels built up inside the glioblastoma cell, while the cells were simultaneously hampered in their defense against it. The term vitamin E refers to 8 compounds in nature, 4 tocotrienols and 4 tocopherols. The tocotrienols alpha, beta, gamma and delta might help to fight cancer and inflammation. They target many molecular paths used by cancer, including survival mechanisms, new blood vessel formation, proliferation and invasion. In a lab study, alpha, gamma and delta tocotrienols inhibited the growth of human glioblastoma cells, and they caused DNA breaks. Delta tocotrienol killed the cells more effectively than alpha and gamma. The delta tocotrienol also worked well in combination with the extracts from Tabernae montana corymbosa, a plant traditionally used for cancer treatment in Bangladesh. In vitro tocopherols showed anti-glioma effects by controlling the cell cycle progression in the glioma cells. In this study, the gamma tocopherol was found to exhibit the most potent and specific control over the glioma cell's progression through the cell cycle. Similarly, another in vitro study also found the gamma tocopherol was more potent than alpha tocopherol in inhibiting the proliferation, adhesion, and migration into human glioma cells. Ellagic acid is a natural compound, and it's found in many fruits and plants, and it may also have health benefits for glioblastoma patients. In general, ellagic acid has been studied for its ability to lessen the spread of cancer to distant organs and to reduce the production of new blood vessels. In human glioblastoma cells, the ellagic acid inhibited the viability and the proliferation of the cells, and it damaged their DNA. The authors then confirmed these results with mice who had glioblastoma, and they found the ellagic acid inhibited the signaling pathway involved in the cancer cell proliferation and invasion. Another study reported the ellagic acid dramatically reduced the levels of proteins that protect the tumour cells from death. A plant extract with ellagic acid killed human glioblastoma cells by regulating the genes involved in cell death. When the glioblastoma cells were treated with the ellagic acid combined with temozolomide, they exhibited a reduction in the invasive and angiogenic capability of the cancer cell. Ellagic acid might also complement another approved anti-angiogenesis drug for glioblastoma. In a cell study, Ellagic Acid combined with bevacizumab led to a reduced viability of cells. In the same study, the Ellagic Acid led to the down-regulation of MGMT expression, and this increases the glioma cell's sensitivity to the cytotoxic effects of temozolomide. Chlorogenic acid is a polyphenolic compound and is found in coffee, green tea, apples and pears, and it has promising anti-cancer potential for many cancers, including brain tumours. The compound inhibited the growth of glioblastoma cells, and it reduced the growth of glioblastomas in mice. Some of the immune cells in the tumours of these treated mice were changed to a form that can more readily destroy tumour cells. Another study found chlorogenic acid inhibited cell migration and the secretion of a protein that's implicated in tumour invasion. In a double-blind, placebo-controlled trial, chlorogenic acid improved cognitive function in healthy participants. This is intriguing given the cognitive difficulties that are common during and after the treatment for glioblastoma. Panax ginseng root contains compounds called ginsenosides, and one of the ginsenosides in particular, R3, has been shown to have an additive anti-tumour effect when combined with temozolomide in an animal model of glioblastoma. In another study using several glioma cell lines that were resistant to temozolomide through their production of MGMT, the R3 was able to block the MGMT production and to reinstate the sensitivity of the cells to the temozolomide. The study also showed the ginsenoside R3 prevented the changes in the glioma cells that allow them to invade the nearby tissue. It appears that at least some of R3's anti-glioblastoma properties are via its antioxidant properties and the induction of senescence in the glioma cells. The R3 ginsenoside is being studied as an anti-cancer agent due to its well-defined role in blocking new blood vessel development in many types of cancer, including the gliomas. Olive leaf extract exhibited anti-proliferative effects in glioma cells. Experiments on glioblastoma cell lines suggest the olive leaf can prevent the differentiation ability of the glioblastoma stem cells. The anti-glioblastoma effects of the olive leaf extract are seen when used alone in a high dose, and it has an additive effect in stopping the glioma cell growth when it's used alongside with temozolomide. The whole leaf extract has generally been studied, and single compounds within the olive leaf, such as oleuperin, also show promise. Black cumin or Nigella sativa seed has been used in traditional herbal medicine for over 2,000 years. While whole seed extracts have shown effective medicinal qualities, the compound thymoquinone in black cumin has garnered the most interest as an anti-cancer compound. The thymoquinone has been shown to affect dozens of pathways inside the cancer cells, including those that are involved in several hallmarks of cancer: immortality, invasion, metastases, apoptosis, and cellular division. The anti-glioblastoma effect include increasing the cell death and creating cellular stress within the glioblastoma cells. The benefits of the thymoquinone may also extend to preventing ongoing brain-related symptoms, as the data suggests neuroprotective and anti-seizure effects. Nicoliside is another anti-glioblastoma compound of interest from the black cumin. This compound is thought to be responsible for the traditional use of the black cumin seeds in treating parasites and worms in the gut. The nicoliside is also approved as an anti-parasitic agent worldwide due to its reliability for killing human worms such as tapeworms. In one experiment, the combination of thymoquinone and nicoliside was able to lessen the invasive properties of the glioblastoma cell model. The combination also had a similar effect in a rodent model that used implanted human glioblastoma cells. For more on herbs, supplements, and natural treatment plans, check out my website.